Hello guys, welcome to my channel. Today I will discuss about cell cycle and its different phases in detail. Watch my previous videos where I have discussed upon GPCR, RTK signaling pathways and science behind coronavirus in detail. Links of the videos are provided in the description. So let's start. The most fundamental characteristic of all living organisms is reproduction. All cells reproduce by dividing into two daughter cells after completion of each cycle of cell division. For example, repeated cycles of cell growth and division results in the development of a single fertilized egg into about 10 to the power 14 cells that make up our body. So, what is cell cycle? Cell cycle involves a series of events which take place in a cell that results in the duplication of the genome, that is DNA, and then division of the nucleus and cytoplasm to produce two daughter cells. In eukaryotic cells, a series of protein kinases regulate the progression of cells through the cell cycle. These protein kinases have been conserved from yeast to mammals. In higher eukaryotes, this cell cycle machinery is itself regulated by the growth factors. These growth factors control cell proliferation and regulate the cell cycle progression depending on its presence or absence. In case of cancer, the cell's dependence on growth factors for cycle progression is lost. As a result, the cells continue to divide, that is, uncontrolled cell growth, which leads to tumor formation. Actually, defects in cell cycle regulation are a common cause of the abnormal cell proliferation leading to cancer, which I will discuss in my cancer video. So, now I will discuss eukaryotic cell cycle in details. The cell cycle consists of four important processes. These are cell growth, DNA replication, distribution of the replicated or duplicated chromosomes to the daughter cells, and finally division of the cell into two. These processes are divided into four discrete phases. Progression from one phase to the cell cycle to the next is controlled by a number of cell cycle checkpoints. These checkpoints check whether all the events completed properly in the previous phase or not. If yes, then the checkpoints allow the cell cycle to progress to the next phase. I will discuss about cell cycle checkpoints later in detail. Now, coming to the phases of the cell cycle. The human cells take approximately 24 hours to complete one cycle. The cell cycle is divided into two major phases, that is the M phase or mitosis and interphase. The M phase lasts for only 1 hour, during which both the karyokinesis, that is the division of the nucleus, and the cytokinesis or the division of the cytoplasm takes place. The rest of the time, that is about the remaining 23 hours, the cell remains in interphase. During interphase, both cell growth and DNA replication occur in an orderly manner to prepare the cell for division. The interphase is divided into three phases. These are G1 phase, S phase, and G2 phase. So, the whole cell cycle is divided into four phases. These are G1, S, G2, and M. Now, let's understand these phases in detail. First, G1 phase or gap 1 phase. This phase corresponds to the phase between the mitosis or M phase and the DNA replication phase. During this phase, the cell is metabolically active and continuously grows but does not replicate its DNA. So, the DNA content of the G1 cell remains 2N in case of a diploid organism. Presence of growth factors are essential for the cells to grow in size and the phase to continue. The second phase is S phase or the synthesis phase. After the G1 phase comes this phase where DNA replication takes place. It is the only phase in cell cycle where the genomic DNA is replicated. At the end of this phase, the genome content of the cell doubles, that is from 2N to 4N, which allows cell division without reducing the genome content from its parent cell. Due to this replication or doubling of genome phase, mitosis performs equal division. After DNA replication comes the third phase, that is the G2 or the gap 2 phase. During this phase, different proteins are synthesized that will be required for mitosis. Here also the DNA content of a cell remains 4N. And finally, during the M phase, separation of the daughter chromosomes occurs and ends with cytokinesis or division of the cell into two, with each having 2N DNA content. Each of these two cells can again enter the G1 phase and continue the cell cycle process. For different types of cells, the time of cell division is different. For example, in case of a human cell that divides in 24 hours, the G1 phase lasts for 11 hours. S phase lasts for 8 hours and the G2 phase lasts for 4 hours, which results in the M phase lasting for only 1 hour. Other cells such as budding yeast takes only 90 minutes to complete the cycle. Here it is important to mention that G1 phase is the longest phase of the cell cycle. On the contrary, some cells do not divide in adults, such as the nerve cells. Some cells divide occasionally, such as skin fibroblast, and these cells enter into the G0 or the quiescent phase from the G1 phase. When needed, these cells can come out of the G0 phase and again enter into the cell cycle. I have discussed about G0 phase in the next slide. By seeing a cell, it is not possible to determine in which stage of interphase the cell is in. For this, it is essential to check for the DNA content in the cell. The process involves selection of a population of cells. These cells are then labeled with a fluorescent dye that binds the DNA. The cells are then passed through a flow cytometer or fluorescence activated cell sorter, that is FACS. This machine measures the fluorescence intensity of the individual cells. More the DNA content in a cell, more the intensity of fluorescence. The data are plotted as cell number in y-axis versus the fluorescence intensity of each cell in x-axis. The distribution shows two peaks. One for the cells with DNA contents of 2N and other for the cells with DNA content of 4N. Here we can see before the replication phase, the cell is in G1 phase where DNA content is in 2N or a diploid cell. During replication in S phase, the genome is doubling, thus the DNA content will be between 2N and 4N. And after S phase and before the completion of cytokinesis, that is G2 and M phase, the cells will have 4N DNA content. From the plot, we can see that clearly. So by studying the DNA content of a cell, we can identify which phase the cell is in. Now, I will shed light on how the cell cycle is regulated by cell growth and extracellular signals. Extracellular signals from the environment as well as internal signals regulate the progression of a cell cycle in defined phases. For example, growth factors regulate the progression of animal cell cycle. Defined cellular processes such as cell growth, DNA replication, and mitosis all must be coordinated during the cell cycle. This is accomplished by a series of control points that regulate progression through various phases of the cell cycle. One such major cell cycle regulatory point was first discovered in the budding yeast, that is Saccharomyces cerevisiae, which is called start. If the cell cycle passes this start checkpoint, the yeast cell becomes committed to enter the S phase and complete one cell cycle. But crossing this start checkpoint is not easy. Availability of specific nutrients and appropriate cell size is essential for this checkpoint to cross. Without these, the yeast cell cycle becomes arrested and enters a resting state. Similarly, in animal cells, there is restriction point, which is analogous to start in yeast. Similar to the yeast, the crossing of restriction point in animal cells make the cells committed to complete one cell cycle. However, crossing of this point is regulated by the presence of growth factors and not nutrients. As I have mentioned earlier, we can see here in the absence of growth factors, the cells enter G0 quiescent phase where they can remain for long period without proliferating. During G0 phase, the cell does not divide but remains metabolically active. When growth factors become available again, they can again return to the G1 phase and continue the cycle. In addition to the G1 phase, some cell cycle can also be controlled at G2 phase, such as in oocytes. Vertebrate oocytes can remain arrested in G2 phase for a long time, such as several decades in humans, until their progression to M phase is triggered by hormonal stimulation. So today I have discussed upon the general idea about cell cycle, role of its different phases, how extracellular signals regulate these phases, and about restriction point. In my next video, I will discuss about cell cycle checkpoints and the role of different cyclin CDKs with emphasis on MPF or maturation promoting factor in detail. To learn in detail about GPCR, RTK signaling pathways, and science behind coronavirus, please watch my previous videos. In the meantime, don't forget to like and share my videos and also subscribe to my channel. Thank you, stay home, stay safe, and study online.